Okay.
It's more like a pseudo discrete.
Um, which Hayworth projection represents the galactose derivative shown in figure 3. So we have that there. Let me see. The Yeah, I'm looking up conversion between Hayworth and chair, and
It's usually the other way. Like it's usually the I've never seen from like
Mhm.
chair to this. It's usually like something else, like those lines or whatever.
Mhm. I think let's see. So what I see is, let's see. Um, down. Oh wait, I'm looking at the wrong one. This one is up, down. The third one, if it's equatorial, is up. And then the one after that, if it's axial, it should be up. And then, yeah, up. Okay. Then this one, perhaps it's down. And so up, down, up, up. Oh, I guess this one should be up for that over there. So for this chair configuration, I think it alternates like so here this OR group is equatorial and we consider that to be up. And then the carbon 2 hydroxyl group is also equatorial, but since it alternates, it's going to be down. And then the third hydroxyl is equatorial, but since it alternates, it'll be up. And then the fourth hydroxyl here is axial. And so if it was equatorial again, it would alternate and become down. But since it's axial now, it'll be up. And then for the sixth carbon over here, it's equatorial. And if it was, and since it's equatorial, I believe it's going to be up again. So I think what happens with the chair that so it'll be like let's see how we can put this. So here in the first carbon, if it's equatorial, it'll be up. If it is axial, it'll be down. And then for this one, if it's equatorial, it'll alternate from if the previous one was equatorial up, then this one would be equatorial down. Oops. But if it was axial, it would be up. And then for this one, if it is equatorial, it'll alternate from the previous one, which if it was equatorial was down. This time, if it's equatorial, it's up. But if it was axial, just like the previous axial was up, this time, if it was axial, it'll be down. And then this carbon here, if this is equatorial, since the previous one, if that was equatorial, it's down. I mean, sorry, previous one, equatorial is up. This time, equatorial would be down. And previous one, if it was axial, it'll be down. So this time, if it's axial, oops, it'll be up. So, so I think how it would work is, um, yeah, it is that it alternates.
They alternate, yeah.
Yeah. I remember something like this from when I took Orgo um that they alternate like that. So
Okay.
yeah. So this could be um tricky here. So yeah,
Yeah.
well alternate. Here's a I'm gonna put an image in the chat. Uh-huh. So you could see that in this chair conformation on the left, on this first um carbon's hydroxyl group, it's axial. And so if it is axial, like I kind of wrote earlier, like over here, kind of hard to see, but if it was equatorial, it would be up. If it's axial, it'll be down over here. So here it's
Okay.
axial. This is axial, so it'll be down. If it was, equatorial like this, it'll be up. And then the next one, if the next one is equatorial, it'll be since the first one, if the first one was equatorial, it'll be up. This second one, if it's equatorial, it will be down.
Okay.
And if this first carbon axial is down, the second carbon, if it was axial, would be up.
Okay.
Okay. So
So this right here is considered down?
this is so okay. The previous one, if axial would be down, if equatorial would be up.
Okay.
Second one over here, if it's equatorial, it'll be down because it's alternating from the first one. The first one was equatorial, it would be up. So this time, if it's equatorial, it'll be down. Just like the first one, if it's axial, was down. Second one, if it was axial, would be up.
Okay.
And then the third one, if it's equatorial, if the previous one was equatorial, it was down. So the third one, if it's equatorial, it's up. Previous one, if it was axial, which would have been up, this one, if it was axial, would be down.
Okay.
Okay. And then the next one, since the previous one, if it was equatorial, is up, this fourth one here, if it's equatorial, would be down. But if this one, the third one, was axial, it would be down, right? And then
Yeah.
this one, if it was axial, it would be up.
Okay.
So yeah, you just alternate like that. But yes, it's
Okay.
easier to do it from the Hayworth to the chair because here we could see, okay, this is axial, which means it'll be down. Here, this is also axial, which means that it will alternate from the previous axial, which was down. So this time it'll be up, okay? And then here it would be oh wait, sorry. I forgot that in a Hayworth there is no equatorial. It's really just up, like above it, or down
Yeah.
below it. So here, this one we know is down, and then this one is down, but it would alternate. So this one would have to be like an up, or rather the other way around, I should say, with with the first one, if it's down, we consider it to be down. And the second one, let's see, actually. This one. Okay, so if this one's actually down, it's down. This one, if it's down, it's up. And then this one, if this one was down, it would be down again, but this one is up. So I believe that would be down again. So I think that, yeah. So I think that it's okay. You know what? My mistake here. So the Hayworth projection is going to, I actually don't think it'll alternate. I think it's just this is down, this is down, this is up, this is down,
Yeah.
this is up, right?
So only the chair ones alternate.
Correct. So
Yeah.
first one's down, and so for the chair, that's the axial down.
Yeah.
Second one is down, and it alternates. So then this one, in order for it to be down, for the second one, it'll have to be equatorial. And then the third one is up. So this third one, in order for it to be up, if the previous one axial was down, sorry, the previous one equatorial is down, this one equatorial will be up. And then the fourth one is down. So since this one equatorial is up, in order for this one to be down, it must be equatorial. All right, and then this one, the last one here is up. So that means that this one would have to be, if the previous equatorial is down, this last equatorial would be up.
Up. Okay.
Yeah. So
All
that could
They
get,
just kind of alternate that. Okay.
yeah, they alternate in the chair, yeah.
Okay. Okay. I kind of struggle with like lenses and stuff. I know the formulas, but I just don't really know how to apply them.
Mm-hmm.
Like I know like the 1 over F equals 1 over DI plus 1 over DO.
Mm-hmm.
But like I don't really know how to apply it, to be honest. Like focal length.
Got it. Let me take a look at something for a sec. Okay. Yeah. I don't know if you could see my, because I opened up a whiteboard.
No, I can't see. Oh wait, I see it now. I have to click on it separately.
Yeah, because I went over this
Oh, right there to the left.
question here. So I will, I guess.
Oh, it is the same question.
Mhm. Oh, it's not letting me.
I can see it.
Um, yeah, I just want to. Okay, let's go back to your shared screen. All right. So, okay. So in the figure above, an object O is placed a distance 2f from the centers of converging and diverging lenses, both with a focal length f. How does the height hc of the image created by the converging lens compared to the height HD of the image created by the diverging lens. Which one is the converging lens? Which one's the diverging lens?
This one's the converging, and then this one's the diverging.
Good. So, so we have um okay, so we're going to be, using the thin lens equation, right?
Yeah.
So 1 over focal length is equal to 1 over object distance plus 1 over image distance, right? 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 That makes sense?
Yeah, yeah, yeah.
Okay. So now, um, object distance they're saying is equal to 2f, right?
Yeah.
So we could just plug that in for object distance. That way we'll have like terms with focal length.
Okay.
So then this would be 1 over 2f plus 1 over image distance, right?
Yeah.
Now I'm going to subtract 1 over 2f on both sides. So I can get 1 over f minus 1 over 2f is equal to 1 over I, right?
Hmm.
Now, what do I do in the math for the math here?
You multiply the bottom right here by 2.
Yeah. So it'll be multiplying the entire thing by 2 over 2 because you're essentially just multiplying it by 1.
Yeah.
So that's why it's not just 1 over 2, it'll be 2 over 2. So this will give me 2 over 2, oops, 2 over 2f minus 1 over 2f. And what's that equal to?
1 over 2f
Yep. And that's equal to 1 over
I.
I. So the image distance is also going to be 2f, right?
Yeah.
Okay. Now, in order to find the size of the image, there's going to be magnification, which is equal to negative image distance over object distance, right?
Yeah.
So the magnification here is going to be negative image distance of 2f over object distance of 2f. This is just negative 1, right?
Yeah.
So what that means is, well, the one part means that it's the same size, meaning the object and the image are the same size, like height-wise. Um, but the negative means that it is an inverted image.
Okay.
And so this is our HC, right?
How is it the HC? I thought it was the M.
So they're saying that HC is the height of the image created by the converging lens.
So is that the same as the magnification?
Oh, yes, yes, yeah. So the mag
Okay.
just tells you the height of.
Oh, okay.
Mhm. So okay, -1 is HC. No, so now let's do it for diverging lens. Now, here, I'll actually put this stuff in like red to indicate it's important, and then this as well. Now, for a diverging optical device, the focal length must be negative.
Negative.
Okay. So we did the math here, it would just be 1 over negative focal length is equal to, the rest of the thin lens. So 1 over object distance, which is 2f, minus 1 over image distance, 1 over I. And we do the same thing. We can subtract both sides by 1 over 2f to get 1 over negative f minus 1 over 2f, and we do the same thing, multiply this by 2 over 2. So we can get 2 over negative 2f minus 1 over 2f is equal to the 1 over I. Um, what do we do? What do we do now?
So it's like a -3
Yep.
because it's -2 minus 1.
Good, good, good. So this image distance then would be negative 2f over 3, right?
Wait, how'd you get the image distance again?
So if this is equal to 1 over image distance
Okay.
would just be 1 over that.
The reciprocal. Okay, I got you.
So, okay, so that's our image distance here. And then for the magnification, which is negative I over O, would be negative negative 2f over 3 divided by 2f, right? And so the 2f's will cancel, and the negative of a negative would give you a positive. So this will give you a positive 1/3, right?
Yeah.
As our HD, right?
Hmm.
Okay. And now it's positive because it's an upright image. And also, I'll write this in red. Um, Image less than 0, aka negative, is inverted, but also real. Real images will be inverted, whereas image
Okay.
is, oh, sorry, my bad. It should be magnification. If that is less than 0, it's inverted real. If it's greater than 0, it's upright virtual. Okay. So we have our HC here and our HD here, right? And they're putting it in terms of HC is equal to some, HD.
Yeah.
So now what we can do is we could um, solve for HC in terms of HD, right? Right.
Yeah.
So that could mean, so let's see, we could do, okay, negative. So if HC is -1 and HD is 1 over 3, we could rearrange, I would rearrange HD. So I'll do it like, okay, if HD is equal to 1 over 3, I can multiply 3 on both sides to get 3 times HD is equal to 1, right?
Yeah.
And if HC is equal to -1, it would just be like this 3HD is equal to 1, but just on the negative side. Um, uh, but since we're being asked about just the heights, we're not being asked about, if it's inverted or, upright. The
Yeah.
We could kind of just leave it as 1 instead of the -1 to get,
Hmm.
and so this would be closest to that, right?
Yeah.
So
A lot of calculations for them.
yeah, yeah, yeah. So something that can also help though is that um so there's this optics kind of like table that I have like in the PDFs folder that can help. Um yeah, it'll just be called optics. Yeah. So here, so actually go back to that other thing because I'm going to just have you maybe take a, oh, just, I mean, in the other tab, just because I want you to take a screenshot of this and then I'll erase
Oh,
it.
I already took a picture of it.
Okay.
Yeah,
So
yeah, yeah.
this optics table is going to really help out. And this would be something that I would actually write in like the first 15 minutes of the, tutorial part of the exam on my scrap paper.
Okay.
Shows that if it's a diverging optical device, it will always, at no matter what distances it is, it'll always produce SUV, which is small upright virtual.
This is for mirrors and lenses. Okay.
So if it's diverging, it will always be small upright virtual. Okay. And then for the converging, it depends on the image distance. Oh, sorry, it depends on the object distance, right? So now if we go back to the that problem, I think it was UWorld. So here, the diverging the diverging um, lens here will always be producing a smaller image, right? So we know that
Yeah.
D must be smaller than HC. Um, oh, and then you could also see in the PDF. So we know the object distance is 2F. So yeah, in the PDFs, if your object distance is 2F, your image distance is also 2F, and your type of image is same size inverted real.
Mm.
So yeah, we go back to the UWorld one.
Okay.
So now we know that for the converging, if this is 2F, the image is going to be at 2F as well. Okay. So right away, so since we know that the height of the diverging is going to be smaller, because if this is 2F, then this is going to be same size inverted real, right?
Mhm.
This one would be SUV, small upright virtual. So we know that HC must be larger than HD, right?
Yeah.
So A says HC is 1/3 of HD, right? And
Yeah.
that wouldn't work because HC is larger than HD, right?
Hmm.
Because if you rearrange this, you would get 3 times HD is equal to HD.
Yeah.
Means HD is larger than HC, right?
Hmm.
But that is impossible, right?
Yeah.
So
And
that's
then B wouldn't work
exactly,
because they're equal.
exactly. So that's where we're left with either this or this. And that's when we did the calculation.
Yeah.
So yeah, so I would say that optics table would be good to like
Yeah.
memorize. Um, but yeah, uh,
Okay.
we did the thin lens equation, yeah. And I like to ask questions about glasses and stuff as well.
Yeah. It's very popular.
Yeah.
It didn't come up when I took the MCAT, but it'll probably come up next time I take it.
Yeah.
Okay. Uh, Okay. This one is also about like sequences and strands, and then I just kind of get lost in it.
The Okay, so let's talk about what's happening here. So now your see. So your so CDNA was generated and amplified by a PCR for several genes involved in memory formation. The following primer sequence was used to generate the CDNA for NPAS4, which of the following nucleotide sequences represents the molecule from which this CDNA was generated. Okay, so this could be a bit confusing, so I'm going to create some white space here. Okay, so the key to what CDNA is let's say that we have the entire genome of something. Okay. Now, only like 2% of the genome of an animal or whatever actually codes for proteins, right? So that means like 98% of a human, like a genome, a person's entire genome is not relevant, not useful. So that's why cDNA is created, because if you think about it, so let's say we start with our DNA and we want a particular gene. So I guess for this one, it's the gene for NPAS4. So we would transcribe that into the RNA or the mRNA for a specific protein that we want. And, the mRNA is going to undergo like splicing out the introns, leaving in the exons. And therefore, we would get the parts of the DNA that's actually useful, like the exons and stuff. So then what we can do is we can reverse transcribe the mRNA into cDNA. And this way, it's only the parts of the DNA that matter. So have you ever heard of like bioinformatics?
Yeah.
What have you heard about it?
It's like something you put in a computer and it's like it gives you this like certain
Mhm.
like I don't know. I took a class that we did a lab about it. But
Oh, cool. You did like blast and
and then you like see the sequences and all that.
the FASTA files.
Fast, yeah.
So like the human genome um FASTA file is like 40 gigabytes, which is
Mhm.
huge. But the cDNA for that is like maybe like 4 gigabytes because it's just the
Okay.
coding parts of the um DNA, right?
Yeah.
So So the actually I think it was like more so like 140 gigabytes large.
Hmm.
Um now so the first step, you take your DNA coding strand and you transcribe that into mRNA. And then you reverse transcribe the mRNA back into DNA, and now that DNA is called the cDNA. Now, there's two types of primers here that can be kind of confusing. So the mRNA, like the mRNA that you get, you are so let's say that we just had like so like let's just say that we had like something like something like this. I'll just leave it like this. So what happens first, or I'll write it down using text. Oops. Yeah. I'm just going to use like text here. So let's say that we have and this is oops, read to 5 here as our mRNA. Um, so okay, so in order for us to like be able to make a new strand, we need to have a free 3' end. Okay. And that's just because the 3' end is when you have your oops. So 1, 2, 3. The 3 is where that hydroxyl group is. And so for the next one, which is attached to, phosphate, this can nucleophilically attack the phosphate, and that's how we can add that next nucleotide in.
Yeah.
So we need to have a free 3' end. So for this mRNA strand here, we have, well, we have a poly A tail here. And so you can use an oligo DT primer, which is going to be, 5 to 3, and it'll just be like T, 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 T. That's the oligo DT primer, and that gives this, a 3' end, which means that then the actual like polymerase can start to fill in the nucleotides for this. All right. And then you'll end up with your cDNA. And since DNA, including cDNA, is supposed to be double-stranded, what happens is this mRNA gets degraded. We don't need it anymore. And now we need to create the 3 to 5' end of the cDNA sequence for it to be a double-stranded cDNA. So, in other words, what that means is that, so I'll use blue. So this is your cDNA primer, and this is your mRNA sequence that is complementary to the cDNA primer. Okay. So, and in other words, I should say that the cDNA primer here is going to be having the same sequence of, or as the DNA like template strand that you start off with in order to make that mRNA in the first place. So this DNA, it'll be like the coding strand of it. And, so let's say the coding strand is like, I use text again. Whoops. Let's say the coding strand is 5' AG AGC, CTT, CGG, CTG, whatever. 3 prime. The mRNA must be complementary to that, or rather, sorry, the mRNA will be coding strand, and then there would be the, sorry,
Template,
template, yep, which would be the
3 prime,
C,
uh,
GAA, GCC, GAC, 5. I think it'll look like that. So this is coding,
template,
template. And then the mRNA, I'll make it in red, is going
and
to
then
be,
the complementary mRNA
yep, it's going to be complementary to the template,
to
which,
the top one, the coding one. Yeah.
well, it'll be complementary um, to the template, which means it'll be essentially the same thing as the coding strand,
Oh
because
yeah.
the coding strand is complementary to the template, and then the new mRNA strand will be complementary to the template, which means the mRNA strand will be exactly like the coding strand. Just be AG,
AGC.
CUU, CGG, CUG, 3 prime, right? Right.
Yeah.
And then your, make it in blue, your CDNA would then be TGC, GAA, CGG, G, or rather, sorry, um, C, TGC, GAA, C, G, what am I doing? TGC, GAA, GCC, got it, okay. Then GAC and 5. That's your um primer, CDNA primer, and that must bind to the
This is the primer, not the mRNA.
correct.
Okay.
That'll be your um sorry, so sorry, this would be the CDNA, and then the primer must be complementary to the CDNA, so that would be like AGC, CGT, CGG, CTG, oops, 3 prime, and this would be the CDNA primer. So you could
Mm.
see that the um the coding strand is basically the
I know
same.
I know how to do these top three, but then like these I didn't know like what they were
Mhm.
talking about.
Yeah. Yeah. So this is where, so this is just transcription, and then from here to here is your reverse transcription to get your cDNA, and then like the,
Mm.
and this is when you use the um oligo DT
And TPs.
primer, but that's different from the actual end result cDNA primer that we get. So that will be the one that would help this new cDNA one form.
Okay.
So yeah. So basically your primer sequence is going to be complementary to the mRNA sequence, which means it'll have the same sequence as the um that means that the mRNA sequence, this one, must be complementary to the cDNA primer, um, which could be like that one. So our primer is going to be, so here they're giving us the primer here, and they're asking which of the following nucleotide sequences represents the molecule from which
Oh,
it's generated. So we know cDNA is generated from the mRNA. So we can get rid of these because they have T's in it, right?
yeah,
And then it comes to, something like this. But you see how it's like the 3' to 5'? And
yeah.
and this part that I talked about with the oligo DT primer, when this gets filled out here, that'll be um your cDNA primer.
Mhm.
So you want to have that. So you see this is the poly A sorry, the poly A tail,
Yeah.
and that's why you want, you know, this will be your oligo DT primer, and then the rest of it will be the cDNA primer. You can see how it's this over here is the oligo DT primer, and then the rest of this is the cDNA primer sequence.
Okay.
And then
So if that's the poly A tail, like what's an example of is this the 5' cap?
it's a little weird here because this is the um cDNA primer, right? So
Yeah.
it's going to be complementary to the mRNA. So, but here you could see that because the poly A tail is on the mRNA and it's on the 3'
Mhm.
end, right?
Yeah.
You have the 5 cap and you also have the poly A tail. The 5 cap is not composed of like really like nucleotides. It's just composed of like, you know, the let me see here. The 5' cap is like, yeah, I'm pretty sure it's like it's like something that isn't composed of nucleotides. It's like some type of methyl thing here
Okay.
in the image. Whoops. That I just sent. Or actually, no, wait, the second one is going to be better. It's like this one that I just sent right now. So yeah, so you see the 5' cap is this entire thing. So it's just this like methylated nucleotide and stuff like that. So it's not really like
Mm.
a sequence.
Yeah.
The poly A tail is a sequence of a lot of A's, and that's why in the mRNA complement here, you have the poly A tail on the 3' end, right?
Mhm.
And so when you make the cDNA primer, this part is the oligo DT primer, and then the rest of it makes that cDNA primer. So
Okay.
yeah, we can go back to that question. And so they're giving us the primer to generate the cDNA. And so this is our cDNA primer. And it's going to be complementary to the mRNA. And since they're asking like the molecule from which the cDNA was generated, it was generated from over here, you could see from the mRNA, right?
Hmm.
reverse transcriptase. So right away we can eliminate A and B because those are DNA. And then we want to recognize that our CDNA primer is going to be complementary to the mRNA. That
Yeah.
so that's this one. Is this, if you just put it underneath here, be complementary, right? Three and then the four A's would be complementary to the four T's here. Then AAG, UUG, C, right?
Yeah.
So there,
Makes sense. Mhm.
yeah.
Or you can just kind of rule out these because it says which one was this complementary DNA generated from. You just say mRNA, so it has to be at least used.
Yep, yep, yep. And
Okay.
so, yeah. And this was apparently tough because 50% chose A.
Mhm.
And you could see that A is just like C. It's just that A, this one is DNA.
DNA, yeah.
So that's that. What's happening there, yeah.
Yeah. Okay. Cool. Okay. It's a physics question about like speed, and I'm not really sure how to go about this question or like draw it out. I'm not sure.
Um, so okay. So I think we could do this maybe without that. So let's see. A tornado simulator uses a motor with a fixed power output to drive a dolly and propel debris horizontally towards a structure at constant speed. Which of the following will occur if the frictional force on the dolly's wheels is increased? Um, so constant speed means like what's the implication of that?
Um, like it's going at the same pace.
So if it's a constant speed, it means that it means what about like tell me about its acceleration.
It's acceleration. It's like zero.
Good. And acceleration is a consequence of a net force with F net equals ma, right?
Yeah.
So if the acceleration zero, the net force should be zero, right?
Yeah.
Okay. Now, if there's friction, okay, friction will move in, will, a frictional force would be in the opposite direction of the object's, whichever direction the object is moving. And if there's a frictional force, that's actually a non-conservative force, which means that it can change it. You're not going to have conservation of energy. So, so yeah, if we have some object moving at constant speed and you increase the frictional force, that's going to give you a net force in the opposite direction, which is going to give you an acceleration that's no longer zero, but it's going to be negative or something in the other direction. And that's going to contribute to the speed of the dolly decreasing.
What does it mean? Like the friction force
Like,
is increasing.
yeah. So, like, this is a box or something, and we are applying a force, let's say force applied in this direction. And if our F net is zero, it means that our acceleration is zero, which means that this net force, oh, and then like, let's say that opposite to this is the frictional force. So if the applied force FA is equal and opposite to the frictional force, our F net will be like FA minus F friction, which gives us a value of zero, which means that the frictional force and the applied force are equal. But they're asking what happens if you increase this frictional force value? Well, if you increase the frictional force, then meaning from initially when they're equal, right? So let's say that I'll use blue. We change this so we have a frictional force that is larger than the applied force. Then your F net, which will be, FA minus F frictional, it's no longer going to be zero because the F friction is larger. So it'll be some value that's less than zero, like a
Oh,
negative value.
okay.
And that would be a negative acceleration. And that means if it's a negative acceleration and we take, the speed or the velocity to be positive direction, it's going to work against it. So it's going to decrease that speed.
Okay. Okay, this is OChem aldol condensation. I don't really know how this like mechanism like works, to be honest. I know it's like shown in here, but I don't really know what it like means. Like what site will be deprotonated, or I know like an enole is like something like a double bond. But yeah.
Yeah. So, okay, we have this molecule, we have this molecule, and then it ends up combining the two, right? So
Yeah.
we want to know where it would combine the two. So let's see at what's different here. Well, the R1 and then this carbonyl part is the same. And then here, the R stuff stays the same, except we lose something like that. And remember, a condensation is just dehydration synthase.
Dehydration. Yeah.
You're going to lose an H2O. So what seems to be happening is this. Oh, and then the first step is going to be deprotonating the alpha hydrogen, which means that this CH minus here, or in this question, this can get deprotonated to give you CH2 minus. That would happen here. And then it can add or attack this carbonyl carbon over here and form a bond with
Um,
it and kick it off to get this. So that first step, yeah, see how it says the first step, they're asking which is going to be deprotonated by a base, right? So if it was this, then this R1 should be the one that that would attack,
yeah.
but it seems like it's not. It seems like it's attacking from this one, right? So,
So is it always, you said the alpha,
yeah,
like
alpha carbon, which means like so like if you have a carbonyl, you have like this one is the alpha,
this one now?
this
Beta.
this is like the delta. So yeah, so this amine group here is not near a carbonyl group. So it's not going to have,
Okay.
because the carbonyl group makes it so that this carbon here has its electron density pulled against, like towards the oxygen. And if it has less of an
Hmm.
electron density, it's going to have a partial positive. And if it's a partial positive, it's going to be likely to get deprotonated.
So in an aldol condensation, it's always the alpha carbon, or
Yep,
over
yep.
here it gets deprotonated first, and then they use a base all times.
Mhm.
Okay. All right. Okay. So overcomplicated that one.
Mhm.
Okay. I think this is another limiting reagent question that I keep missing.
Mhm.
Uh,
Yeah. Let's see. Okay. When 10 ml of solution, or let me see. Okay. Solution one was prepared by, okay, doing that. Okay. So if 10 ml of solution one and 10 ml of solution two are mixed, what is the mass of the silver sulfate? That's what they do here. Okay. Produced in the reaction. Assume that the conversion of the limiting reactant goes to completion and everything else. Okay. So solution one, they took this with, and they added water to it to make this. Okay. So if we have 10 mLs of this, right? So uh okay. So 2 molar times 10 ml, which is going to be 1 E negative 3 liters, gives me 2 E negative 3 moles, right?
yeah.
And from that, since they give us the gram for the molar mass, we can solve for the mass of that. But I'm going to leave it as moles for this. And then here we have silver, okay, 2 molar. So this is going to be 2 E negative 3 moles of the AgNO3 over there. And then they said
Yeah.
volumes of solution one and two are mixed, resulting in the formation of NH4NO3 and a white precipitate, which is the Ag2SO4. So, okay, now let's see here. Okay, we're gonna have to make a balanced reaction, I believe. So we're gonna have NH4 2 sulfate plus AgNO3 yields NH4NO3 plus Ag2SO4. So we have 2 silvers on the right and only 1 on the left. So I'm going to put a 2 there. And then we have, let's see, NH4. On the left, we have 2 NH4s. And on the right, we just have 1. So I can put a 2 here to get 2 NH4s on that side. And also on the left, I have 2 NO3s. And on the right, I have 2 NO3s. So, and then Okay, yeah, I think this is balanced over here. So okay, we balanced it. And now from the balanced reaction, we see the mole kind of a ratio here, right? Like for every 1 mole of this, you need 2 moles of this, right?
Yeah.
Now we did the math and turns out we have the same amount of moles for each of these, right?
Hmm.
So if we have the same amount of moles, according to this, what would our limiting reagent be?
If we have the same amount of moles,
Mhm. Think about
I don't,
uh
since they're equal.
yeah. Um Meaning what we have is equal amounts, but what we need is, so let's say we had 1 mole of this, we need twice as many moles of this.
Yeah.
But we only have,
Uh,
yeah, we only have the same amount of both, right? Right. So that means that our limiting we need twice as much of this.
so this one would be it.
Yep. So that means that this reaction will proceed until this runs out. Okay. And when this runs out, they're asking how much of this we would produce, right? So that means that we're held back by just 1 mole of this, which is like the point, um, the 2e minus 3.
Hmm.
Oh, wait, hold on. I made a mistake. It was 10 e negative 3 liters. Okay. And that would give us 20 e negative 3, which is just 0.02, right?
Hmm.
Moles. And then this one's also 0.02 moles. So we're making, let's see, resulting in the formation of this and that. So we're making this. And let's see. Okay. Let's run out this much tracks. So, okay, for every 2 moles of the this over here, we have 1 mole of this, right?
Hmm.
So the moles of this should be 1/2 of the moles of this, right?
Yeah.
So then this would be 0.01 moles of this. And then we can use the molar mass to solve for so this is 312 grams per mole of this. And so this can just be like, I could do 312 times 1 e negative 2 to give me 312 e negative 2, which is just going to be 3.12, right?
Yeah.
Mhm.
I think I just didn't know what the limiting,
Got it. Yep.
well, because they were equal at first, and then I just, and then so when they're equal, this one's more because
Because you
this
need,
is gonna be more of it to make.
yep.
Okay. All right. Okay. That makes sense. And I just had one last question on Altius. When I was taking the full length,
Yep.
I think I was talking about discrete signals. And I don't know if this is related to like the conjugation or
Oh, this is just like HNMR.
yeah.
So discrete signals. Okay. So,
Yeah, what does that mean?
oh, it means how many non-equivalent hydrogen neighbors you have, right? So
Okay.
we could see that there's, let's see. So, okay, so we have hydrogens here, and okay, for this carbon, we have the two hydrogen neighbors here. We have one hydrogen neighbor there. We have another hydrogen neighbor there. Here, hydrogen neighbor, here, a hydrogen neighbor. Now, like,
Now let's say, sorry, side note. So let's say there was no double bonds here, so there'd be two hydrogen neighbors.
yeah.
Okay.
Um, so, but yeah, we really want to just see how many unique hydrogens we have. So, for instance, these two hydrogens have no neighbor.
Yeah.
They're attached to a carbon that has no, that's not attached to any hydrogens.
Hmm.
So that'll give you a singlet here, like just like one signal like that. And then for this one, we think, okay, how many neighbors, non-equivalent neighbors does it have? Uh,
One.
or rather, sorry, for this one here, how many, non-equivalent neighbors does it have? So it has the two from the NH2, one over here, but this and this are identical. So this will just give you a uh, a signal for, I guess, one, two, so like a triplet. And then
We're
lastly,
not we're not counting this because it's identical, or
oh, we are counting that, but it's identical to this one.
then we're using the two hydrogens here, or no? Or is this just counting as one?
Oh yeah, so that we're using that as well. So we're basically doing this. So for the, I'll just kind of draw. Okay, I'll start with green. So for, let's say, these hydrogens are just attached to this nitrogen, but that nitrogen isn't attached to any other hydrogens. So this is our first signal here. And the number of non-equivalent hydrogen neighbors is zero, so n is equal to zero, but n plus one will be equal to one. So you'll get one signal, which is just going to be a singlet. Now the next one I'm going to use pink. For now, for this carbon, we want to see, okay, how many, non-equivalent neighbors does this have? So it's attached to this nitrogen that has these two. So it has a non-equivalent neighbor there. And then it's also attached to a carbon that has this hydrogen. So that's another non-equivalent neighbor. It also is attached to this one with one hydrogen, but this one is the same as this one. There's no way to distinguish the two. So that one won't count, and you would just get, these two hydrogens and then this one, where n is equal to 2, n plus 1 would be 3. So that'll get you your next signal, which is a triplet.
Okay.
Last one, I'll use orange for this. So now this has this as a neighbor, but since this molecule, whoops, goes like that, like this carbon over here doesn't have any hydrogens attached to it. So that means that this one in orange just has one non-equivalent neighbor. So that's going to be n is equal to 1, n plus 1 is equal to 2. So you'll get a doublet, but ultimately you get 3. Uh,
Okay.
yep.
All right.
Mhm.
Okay. That makes sense. So just because they're identical, you're not counting
Mhm.
because they're non. Okay.
Yeah, they should be identical hydrogen neighbors. Yep.
Okay. All right. That's all the questions I asked. I just have some questions about our schedule. Um, so I've taken 4 full lengths, third party. I'm supposed to take another one on
Oh,
third or
have you been putting?
Friday.
Have you been put?
Yeah. So the last one I took, kind of dropped in cars. I don't know, I wasn't having a good day, I guess, or I only got 18 out of 52 right. So I'm not sure it's timing or
Mhm.
wasn't feeling good or whatever.
Got it.
But I was wondering if I should take a one of the AAMC exams that I've already taken for this next one to kind of gauge where I'm at because I didn't really review them properly.
So,
But I know because I know I have scheduled for next month full length one, the sample,
yep.
and then
So I think let's meet next to go over like questions from the full lengths that you took.
okay. Well, I kind of already which ones the Altius?
Yep.
Well, I kind of already went over well because they have like
Huh?
what's nice about it is they have like video breakdowns of each one,
So
and I was kind of able to understand them.
are you okay, so you don't have any questions for those?
No, just the ones I went over, like just that one question I have with you.
Got it.
But then for like, let's say if I do take like either a blueprint or an AMC, I probably have to review them because they don't have any like those little videos.
Got it. Got it. Got it.
And I know like the AMC ones are like important to review because
Mhm.
I don't know.
Got it. All great. Yeah. So I mean, I would say,
Or just stick with the third parties for now and then just
yeah,
see how where I'm at. The
yeah. But I would say, yeah, I know that you're reviewing them and stuff. Um, but yeah, I just want to make sure that because if you're not improving that much between the tests, then I think there might be some issue with the review process between the tests. So,
yeah.
like, yeah, maybe we can even do a full passage together or something like that. Um,
Yeah. Well, we can do that.
yeah.
Should
Okay.
we yeah, or like for next time, should we wait for the guess there's a new for AMC, the section bank.
Mhm.
Maybe we can walk through one passage together and see we
Yep.
can like see where my head like where I feel like I'm struggling with like pinpointing what the each passage is talking about. I feel like I know some like the content or at least some of it to the point where like some of the questions are just straight up asking me what's in the passage and then I just don't really know how to break it down.
Got it. Got it.
Yeah. But yeah, that's all I have
All right.
to
Awesome.
say.
Yep. So yeah, it'll just be 113 for today. And yeah, you can send that along. And then I'll see you sometime
was thinking maybe Wednesday,
like
but
this Wednesday.
yeah. Does he have anything available?
Yeah. Does 4:30 work?
Eastern? Yeah, let's do that. Then you said 1:13?
Mm-hmm.